Okay, looks like the Hangout is live. Um, Actually, if anybody wants to go and find the link and just make sure that we're broadcasting, so this definitely gets picked up. That'd be helpful. So the links are in the announcement, one of the many announcements that we've had over the past couple days. But we should be live. That 1 p.m. link from yesterday should be functioning.
Yeah, I can see.
You can see it? Okay, great. Great. Okay, so I am going to switch. So I guess you can't do full screen when you're presenting, but we'll just make this as large as possible. Okay, great. Now everybody turn that off because it's distracting. Fantastic. Okay, so this is the final study guide. As I said last week, basically the stuff that you guys are going to be tested on, just as a review, or some of the things that I mentioned last week. Mixed models, you're not going to have to do proofs. Fine mappings, that was the Caviar paper, that mouse genetics paper from Jonathan Flint. Some stuff on human ancestry that we've talked about in class. Also on population structure, which sort of fits in with mixed models and a lot of the stuff that we talked about. And then also there are some questions on the probability foundations for this class. Basically, you know, to be very clear, there won't be any proofs on the exam period for undergrads or grads besides the people who are going to be doing the derivations again. As part of that deal for people who didn't do so well on the midterm, you'll have the option to retake question 4, that 4-part question that was all the derivations from the midterm on the final. Yeah.
On the video, I can't see the screen.
You can't see the screen on the video. Okay. Okay. And thank you. Okay. Is that okay? Hopefully that's just lag.
So are you recording this?
Yeah, so it's going on YouTube right now.
That's live?
Yes.
Yes. No one say anything stupid.
Yeah. Okay, can people see the study guide on the screen now?
Yes.
Okay, fantastic. Yeah, so
Any audio is working?
you can turn the audio on again for a sec just so we can make sure. That sounds like it's working. A lot of ums, long, luscious ums. Okay. The first thing that's here, this is just something that I didn't put on, that I didn't put in the list of topics that I mentioned last week, but something that I think is important for you guys to know about and to think about when you guys are doing GWAS is why inputs are standardized. So there will be some questions on like the actual doing of GWAS and like what are some of the steps that go into it. So this on standardizing inputs. Why do we standardize inputs when we do GWAS in general? So there was a bit on this in the missing heritability paper from like way back, and then we did it again a couple weeks ago. But why in general do we standardize inputs? What does it even mean to standardize your input? Some people call it normalize, but standardize is probably the better term. To make the math work, that's exactly right. Yeah. And in particular, what works out nicely when you have standardized inputs? Yeah, yeah, that's pretty much it. So the mean of that mean term, if you just use like 0, 1, and 2 for your input genotypes, if you just use 0, 1, and 2, then the mean of that is going to be very different from the actual mean value of the output phenotype. So the basic reason to standardize your inputs is because the math works out nicely, but in particular the mean term in your basically GWAS model is going to be the same as the mean of the phenotype, which you won't have if inputs aren't standardized. There are some other like things related to the math that are useful when you standardize your inputs. So like you always know that the variance is going to be 1. So you have doing like t-test is a little bit simpler, but basically the reason is because the math works out nice, and in particular because the mean in your model is the same as the mean of the distribution. Oops. And okay, any questions on that? Okay, second question on mixed models and population structure. It's a pretty large hint for this question. So give a simple example of a GWAS, like a study, where failing to account for population structure will result in, let's just say, a mistake, where you'll get bad results when you do a GWAS. Can anybody think of a simple genome-wide test that will give bad results if you don't account for population structure? Yeah.
The obese mice, the like I don't know, lab strain versus the wild strain.
Okay, so why does that not work? Or why do you get bad results if you don't account for population structure?
Because there's been like a bottleneck and selected for particular traits. So then like pretty much every variant that's within those will be associated with the trait.
Very good. Yeah, that's a perfectly good example of something. So every gene that's associated with being a lab mouse is also going to be associated with being obese. Every gene that's associated with being a wild mouse is going to be associated with being non-obese. So in that case, you have a huge population structure problem, and that'll hurt the results of your GWAS. Can anybody think of another one? And a hint is that probably they're all going to have to do with population structure.
If the people in your city are related, basically anything that's associated with your results are confounded by the fact that they're related.
Very good. Right. So another thing that you can have is like family relatedness. So if people in your study are related to each other, or if you do like a study in multiple cities and you find that the people in one city are more inbred than in another city, then you might find some associations there as well. That's a good one. Another thing might be very similar to the different cities concept is if you have like 2 studies run on like different machines, that might account for some of the difference. So you might want to take into account the structure of that. But those are all pretty simple and clear examples. I mean, what you're looking for is a very simple like population structure that's simple and obvious, and communicates that you understand why having this population structure can give bad results if you don't account for it. Okay, any questions? Okay, fine mapping. So this is from the Caviar paper. The question here is it easier to perform fine mapping in a region with high LD or low LD, and why? All right. What is LD in the genome? Linkage disequilibrium. Great. And what is linkage disequilibrium?
It's like when you have the SNPs and then like they're correlated, but if they were just like independent of each other, they wouldn't be as correlated.
Good. Yeah. Basically, you have correlations between different SNPs in the genome. So high LD means you have a lot of correlations. There's the correlations between different SNPs are very tight. Low LD, the SNPs are mostly independent, right? That's the basic difference between. In low LD, the SNPs are mostly independent of each other. So if that's the case, if I have like 100 genotype SNPs in a region with high LD and I want to choose a couple of them to test versus 100 genotype SNPs in a region with low LD and I want to test a couple of them, which one of them is it going to be easier to do the fine mapping in? So let's see. So just to jog everybody's memory, oh yeah, go ahead.
Yeah, if you define fine mapping.
That's exactly what I was going to do. Let's jog everybody's memory about what fine mapping is. So we all read the Caviar paper. We all posted about it in the forums. We all learned something from it. What is fine mapping exactly, or generally? So if you do a GWAS, right? What's the most common thing you get out of a GWAS? You get p-values, and then if you plot the p-values on the genome, what is that called? So that's called a Manhattan plot, and it looks something like no. Okay, so it looks something like, oops, like this, right? These are the p-values on the y-axis, and the coordinate in the genome are basically the chromosome and the location within the chromosome that gene is on, or that SNP is on, are on the x-axis. This is an example from the Jonathan Flint paper, where either they do or they don't correct for population structure. So at the top is an image of the chromosomes, or of the p-values when they don't correct for population structure, and they have this huge number of significant SNPs, way more than than is probably true. And definitely way more than you can afford to test. So what fine mapping does, fine mapping in this case, fine mapping would be identifying the SNPs that are the small subset of SNPs that are most associated with, or that are most likely to be the true associations. So if you find a lot of significant p-values, fine mapping is going to give you the ones that are the most likely, actually contain the causal SNP, right? So our GWAS only detects, it's a genome-wide association study. It's not a genome-wide causal study. Fine mapping is the process of identifying potential causal SNPs from a set of associated SNPs. That's what the basic premise of fine mapping is here. And you can see, for example, in figure B on this, in this figure, or part B of the figure, the reason that we have all of these huge p-values is because we have a lot of population structure. But you can also see that there's a lot of LD. So for example, every time that you have a peak, there looks like there's a lot of SNPs clustered around it. Versus, for example, in chromosome 5, there's like a big area of really tightly correlated dense peaks, and that's something that you also see even when you do correct for population structure. You still have LD left over where SNPs are correlated with each other. Okay, so that's fine mapping. Fine mapping is we're trying to find a small set of causal SNPs from a larger set of associated SNPs. Which one of these is easier to do when there is a large amount of LD or when there's a low amount of LD? Which one of these is going to be easier to perform fine mapping in?
Low LD.
Low LD, right? When the SNPs are independent of each other, the peaks are going to be more separated, and it's much easier to identify the causal SNP in that situation. When there's high LD, the SNPs are very good tags for each other, right? So if you want to do like a tag snip analysis, you want LD, or because the high LD is going to give you good tagging, but it's hard to tell SNPs that are good tags apart from each other. Yeah.
Sorry, what is LD?
LD is linkage disequilibrium, which is the degree to which two SNPs are correlated with each other, or kind of any pair of SNPs within a region.
Sort of what you that one for the degree two SNPs are.
The degree to which two SNPs are correlated with each other. So, or importantly, it's like how much they diverge from being independent from each other. So it's two ways of saying the same thing, but it's the sort of divergence from being independent. So, for example, if you have two SNPs that are that both have 90% frequency in the population, if 81% of the individuals in the population have both of those SNPs, that doesn't just because they both have they have 81% of people have both of them doesn't mean that they're really tightly correlated. Actually, that's what you would expect from random chance, right? So 90% times 90% is 81%, but that's the general idea. Okay. Yeah.
I was writing stuff down. I hope you didn't just say this.
Yeah.
Can you read the Y for this? So it's better with low LD because.
Because in a region of low LD, there will be peaks will be, or large p-values will be well separated from each other in general. Or like, yeah, large or strong p-values will be very distant from each other when there's no correlation between nodes or between things. Okay. Any more questions on this? Again, this is a typical question, and as you guys know, like many of these typical questions make their way onto the exam. There may also be some questions with slight modifications, some that are slightly different, but like there's definitely going to be stuff on fine mapping on the exam. So yeah. Okay, some stuff on statistical foundations. This stuff is very important for you guys to, like, for the purposes of job interviews, these are the ones that are probably going to make you the most amount of money just because they're very broad. And if you say them really well, you'll sound very smart. So let's go back to this one, which was on the midterm. Define the term p-value. How do we define a p-value?
Like the probability of getting like what you got under the null.
Pretty good. There's a little bit more.
It's a chance of detecting a higher statistics than the mean value under null hypothesis.
Yeah, that's pretty close. Let me just this is how I define it for myself. It's the chance of finding a value that is more extreme than the observed value, the observed statistic under the null hypothesis. So it's the chance of observing something more extreme than the observed value under the null hypothesis. That is how I define the term p-value to myself. It's how I recommend that you guys also define the term p-value. I think it's pretty short and succinct and kind of captures all the information. Okay. Okay, great. Any questions on that definition? Yeah.
Sorry, it would be just because it's how I remember it. Would it be wrong to say to observe your observation or observation more stream because it's continuous? It doesn't matter.
It turns out that it doesn't matter. So you can because it's continuous, the probability of your observation under the null is actually 0. But so you can only talk about the probability of a range of observations. That's why when you give like a confidence interval, it has to be an interval. It can't just be Can't just be 1 point, because the actual probability of any individual, you know, the probability of like drawing from a normal distribution and getting, you know, 0.5 is 0. Because but the probability of getting a value between 0.4 and 0.6 that you can define, because there's like real probability mass there. Anybody a math major? Okay, great. Have you done like the Lebesgue integral? Lebesgue? Okay. Well, Czar probably knows what I'm talking about. You forgot? Anyway, it's very difficult to take the integral of something that's like really, really narrow. And when you're looking at a probability distribution, unless there's like a jump in the CDF, there's no, like the probability of that individual point happening is nothing. So as long as you have a density that's finite, the probability of that individual point is 0. But that's like, the point is that you don't need to say your point or higher is the same thing as saying high, just strictly higher, because the probability of getting your exact value is 0.
So both are cool.
So what?
Both are okay.
Both are okay, yeah. But the other one's more succinct and makes you sound smarter. Which is why I favor it. Or you could say at least is extreme. At least is extreme is also acceptable, rather than the word more extreme, which is what I use. But they're mathematically equivalent. Okay, define the term statistical power. So what is that? The definition for statistical power is a lot simpler. Let's see. What do we think? How would you define a study's statistical power? Yeah.
The probability of detecting significance, given that the is significant. I don't say that.
That's pretty close. Can anybody maybe revise that a little bit? It's pretty good. It's got most of the right words there.
Chance of detecting a true association under alternative hypothesis.
That's a very good definition. So it's the probability of detecting a true association under the alternative hypothesis. That's a pretty good one. Also, it is the probability of detecting a significant association, given that the association is true. That's my favorite definition. It's the likelihood of detecting a statistical or a statistically significant association, given that a true association exists. There are some more assumptions that are sort of being swept under the rug there, but I think it's a simple explanation that's pretty clear. Statistical power is the chance that you detect a statistically significant association, given that there actually is a true association there. Okay, cool. Let's take a look at this one. Or I'll scroll this all the way to the top. What is the relationship between power, statistical power, and type 1 error rate? So I think it'll benefit you guys to know the difference just right offhand between type 1 and type 2 errors. So what is the relationship between power and type 1 error rate? Yeah.
Type 1 is false positive, and type 2 is false negative.
Type 1 is a false positive, type 2 is a false negative, yes. But even those terms are a little bit tricky. So what is a false positive is rejecting the null hypothesis when it is in fact true, which is a type 1 error. So that is, or in the language of GWAS, a type 1 error is saying that a SNP is significant when it is in fact, when it's not associated, when the association is not true. A type 2 error, also called a false negative, is a failure to reject the null hypothesis when the null hypothesis is not true, or when the null hypothesis should have been rejected. Or in GWAS terms, a type 2 error is not calling a particular SNP significant when there is in fact a true association. So those are all essentially equivalent, and some of them are like domain specific, but you should be able to communicate in all of them. because hopefully somewhere down the line, when you have a job interview and they want to pay you a lot of money for your mathematical skills, you will sound really good when you talk about the difference between type 1 and type 2 errors. Okay, so what's the relationship between power and type 1 error rate and false positives? What? It is not. It is not strictly inverse. This is kind of a trick question.
Yeah, they're not quite related, right? It depends on.
I would accept there is a very limited relationship here as a correct answer. There's a slight relationship between power and type 1 error rate, but through the p-value cutoff. That's right. But there's a much more limited relationship between power and type 1 error rate compared to power and type 2 error rate, right? What's the relationship between power and the rate that you get false negatives? 1 minus, right? So the number of if you have 70% power, that means you have 30% false negatives, right? 100% minus the power is the type 2 error rate, but the relationship between the type 1 error rate and the power is limited, and in particular, like Brian said, the higher your p-value threshold, or you can say that type 1 error rate and power are connected by the p-value threshold. A higher p-value threshold decreases power and decreases type 1 error rate. That's the clearest connection between those two. But really, the connection is pretty tenuous. Yeah.
So when you mean higher p threshold value, does it mean?
I mean a stronger p-value. So a lower, I should be more specific. A higher negative log p-value, a lower actual p-value, or a stronger. What I mean is stronger. Stronger p-value meaning stronger. Yeah.
Wait, no, please. What does that relate to? You said if there's a higher threshold or a.
So if the threshold for determining an association to be significant is a stronger threshold, so the p-value has to be closer to 0.
So when we're running like normally we do 0.05, you mean take it down to like 0.04?
Sure. Right. That would decrease the type 1 error rate because you're going to reject more of the thing. You're going to have fewer false positives. Because you're going to have fewer false positives, but you're also going to have more false negatives because you're not going to accept some of the ones that were true that fell in that range between 0.05 and 0.04.
So it lowers your power.
So it lowers your power and your type 1 error rate. If you have a stronger, which is to say closer to 0, p-value threshold. Yeah.
So it sounds clear. If you reduce the alpha threshold,
Yeah.
you would also reduce the type 1 error rate,
Mhm.
which would increase the type 2 error rate.
Yep.
But we would reduce the power.
Yes. Yep. So if you increase the p-value threshold, some of the things that were significant under the old p-value threshold are now not going to be significant. For the ones that are sort of in between those two p-value thresholds, some of them are going to be true and some of them are going to be false. The ones that are true are going to reduce the ones that are true that you now are now saying fit under the null hypothesis. Reduce your power. The ones that are false that you now say fit under the null hypothesis because you raise the p-value threshold. Reduce your rate of type 1 error, so you get fewer false positives. Okay, there's a lot of factors that influence power in GWAS. Let's see if we can name 4. Sample size, great, big one, easy. What else is out there that influences power? What's that? The p-value threshold influences power, right? We just talked about that. Great. What's that? The effect size, right? A larger effect size is going to is easier to detect. Great.
exactly is effect size? Is that how much this particular SNP is affected?
It's how strong the effect of the SNP is on the phenotype. Yeah.
Allele.
The allele frequency, right? That's another one that definitely affects. So if you have a rare allele, it's going to be harder to detect its influence compared to one that's more broadly distributed in the population. Great. So that's 4, but there's a couple more that we can that you guys that are worth knowing. So not just sample size matters, but the distribution of cases and controls within the sample. So if you have you're going to have your most power per number of individuals when you have equal numbers of cases and controls. Whereas if you have a study that has like 10 times as many controls as cases, you're only going to get like twice the power of having, or about twice the power of having equal numbers of cases and controls. So that's yeah, and then also let's see the distribution of the disease in your population or the phenotype. Okay, and I think that's about it. Can anybody come up with another one? Those are
Population structure, maybe?
That's pretty reasonable. I think we usually think of population structure as affecting more of like the error rate, because like if you have this population structure, you might detect tons and tons of effects, but a lot of those effects are going to be false or are not going to be real. So you'd have to really argue for population structure.
Argue that you would like make your p-value more strict and then.
Yeah, maybe. But I think the biggest ones, and we hit them all, are sample size, allele frequency. So distribution of the dependent variable, the frequency of the disease or the sort of dynamic range that you have of the disease. So the like distribution of the output, the p-value threshold. And what else?
Effect size.
Effect size, very good. Right. So effect size, p-value threshold, sample size, distribution of the allele in the population, and then distribution of the disease in the population. Yeah.
So I'm a little confused. There was 4 different distributions that came up in those. So
Yeah.
you said distribution case control, distribution output, distribution of
Of
disease and the output of it. Are they all the same thing?
the distribution of the output and the distribution of the disease in the output are the same. The rest of them are all different.
So distribution of cases and controls,
Distribution of cases and controls and distribution of output are identical.
identical?
Yeah. Okay. Yeah. Okay, so we saw this in one of the more recent lectures. Let's define these terms. So we'll talk. There will definitely be something on the exam on human genetics, possibly something very similar to what is on the screen right now. But how do we define the terms mitochondrial Eve and Y chromosomal Adam? Who are those people, quote unquote? Yeah.
Mitochondrial Eve is the farthest back female we can identify using mitochondrial data. The current population, Y chromosomal Adam is the same thing, but the Y chromosome.
Yeah, so mitochondrial DNA is inherited strictly from your mother, right? Only the egg passes on its mitochondria to the subsequent generation, at least in our current understanding of things. So mitochondrial Eve is the most recent common ancestor of basically everyone on the planet, but everyone's mitochondrial DNA can be traced back to this woman who lived, I forget, like 180,000 years ago or something like that. I think she's a bit before. She lived in Africa, 180,000 or 200,000 years ago, about. And then Y chromosomal Adam is the most recent common ancestor of the most recent common human ancestor of all males. And this person lived also in Africa, but a little bit later than mitochondrial Eve. Yeah.
So there's, might be a little too subtle for me to understand, like the Y one is only related to males, so we can only, all males are related to Y, but for Eve, everyone is, but only through their mother's DNA.
That's right. So mitochondrial Eve is basically everybody sitting here and in the world has mitochondrial DNA that came from this mitochondrial Eve woman, or we're descended through her. So it's the most recent common female ancestor of all humans, and then the Y chromosomal atom is also the most recent male common ancestor of all humans, but it's for example, it is actually, that may not be the case. So he's definitely the most recent common ancestor of all males because males have the Y chromosome. It may be that there are women that exist that are not descended from mitochondria or from Y chromosomal atom, because their fathers, right? We're only checking by one chromosome, or by basically something that's private to men there. So there may have been women living at the time of Y chromosomal atom who were not, who did not have children with Y chromosomal atom, but those children didn't pass their Y chromosomes on. So there may still be women who are not descended from Y chromosomal atom, but there are not men who are not descended from Y chromosomal atom. Does that make sense?
Yeah, so it's like full populations from Eve through mom's DNA, but all men are from Adam.
That's right. Yeah.
Do you happen to know the mechanism for like mitochondrial? So like sperm have mitochondria, right?
They do, but they're wound up. So like in sperm cells, the mitochondria are like very, very different from what they are in like the picture of a somatic cell that you see. They're like wrapped up in somebody who knows somebody must know the biology better than this, but I think they're wrapped up in like a twisty shape in the like place where the head of the sperm attaches to the tail, and I think that's where they live. Does anybody know the? Anyway, if you look at like a high resolution like electron micrograph of a sperm cell, I think that's what you see. But probably easier to like if you have molecular biology of the cell, or if you don't like borrow it, they have a very good schematic drawing of it. But yeah, the sperm does have mitochondria to eat food and make energy and move its tail, but as far like, and probably someday you'll find out that like some of those mitochondria do make it into the gamete. Probably we'll find that out someday, or we'll find like an example of that happening. But for the most part, mitochondria are strictly the ones that are in the are strictly descended from the ones that are in the egg, the ovum. Okay. Any more questions on that? Okay. Yeah.
Just on the topic of mitochondria, apparently there was a paper in 2014 that found egg cells contain vesicles that seek out and destroy mitochondria.
Oh, really? That's very interesting. So they have one of the many things the egg does to prevent from being over-fertilized, I guess, is to kill the spermatozoa's mitochondria. So very interesting. Okay. The out of Africa hypothesis posits that anatomically modern humans left Africa about 140,000 years ago. If this hypothesis were true, would one expect to find more or less genetic diversity in African populations compared to non-African populations, and why?
More.
You find more genetic diversity in African populations compared to non-African populations. Does that match up with what you see in the like 1,000 genomes data set? That answer is yes. So if you the population with the most genetic diversity is the Yoruba population from Nigeria. I don't know if they have other populations that have higher genetic diversity, but it's very clear that that there are a greater number of single nucleotide polymorphisms in the African population compared to the non-African populations. And what is the reason for that? time, right? There's a big bottleneck getting, leaving Africa. So there was a big decrease in the amount of genetic diversity in the population that left Africa compared to the population that stayed. So as a result, African populations almost uniformly have higher genetic diversity than non-African populations.
Can you repeat that? So people that left here had.
So because when they left Africa, there was the population of breeding humans who left Africa 140,000 years ago was very small in comparison to the ones who remained in Africa. It was a small fraction of that human population. The amount of genetic diversity in that population shrunk by a lot. And then also when you have a small population, there is a lot of drift, for example, or there's a lot of opportunity for there to be drift in that population. And so that reduced the genetic diversity of the population that left Africa and then founded, the populations, the non-African populations of the world.
Hypothetically, if this question is severe,
Yeah.
so cause less braiding and less confusion, could we just say time?
No.
Okay.
That's a little too flip. A little too flippant. You should, if you were to go say the African lineages of humans are older compared to that, but I think you should probably mention the bottleneck effect, and the population of humans that left Africa was likely a lot smaller than the population that remained.
Just understand the bottleneck, just meaning the people that, so I'm confused with the bottleneck first. So you're saying that, does that tie into the other point that the population was so small, the ones that could actually leave?
So the point is that the population of people who left Africa was a small fraction of the total populations of humans living in Africa. So the total human population living in Africa, was probably in the thousands, right? And probably, not more than, maybe, or tens of thousands, and probably not more than a couple, on the first, walk out of, through the desert or across the Gulf of Aden or whatever, wherever, humans first landed when they left the African continent, there's very likely a much smaller number of those people. Okay, mouse genetics. So if I have a mouse population that is the result of 30 generations of inbreeding via brother-sister mating, how many haplotypes will there be in that population? How many would you expect to see? One, right? You would expect to see exactly one haplotype, because after that much time, there's whatever genetic diversity there was is basically going to be replaced kind of in blocks over time. They'll just, by random chance, inherit a whole chromosome from a parent, and by inheriting that entire chromosome, that removes one haplotype, one haplotype at one chromosome out of the population, and over 30 generations, all the people in the population are going to have, the identical haplotypes. So you have one haplotype in that population. Okay, we have, let's see, I think just two more. Okay, great. So this is just the last question then. For grads, I'll give you a hint about what you will see that isn't here. Okay, what are some of the benefits of working with a collaborative cross or hybrid mouse diversity panel population, HMDP, compared to a classically inbred strain? Yeah.
The classically inbred ones are kind of fake. They're fancy mouse that were practiced with pets. They're much bigger, and they don't capture the whole genetic diversity.
That's reasonable, but the general reason that we study mice is not for the mice's benefit. It's for our benefit. So the fact that they're not totally representative of the mouse population to me isn't one of the top reasons to use that. Yeah.
You just have more, like there are fewer like linkages within, like there's less extensive algae.
Okay, and why is that beneficial?
Because you need a recombination event to figure out where, in order to like, because within an algae block,
Mhm.
SNPs are correlated.
Mm-hmm.
So if you have a shorter algae block, then you'll have less like, you're more likely to capture the cosmic SNP as opposed to something that's correlated.
Very good. Yeah. So that's a very good reason for using a collaborative cross or an HMDP population, because they will have they'll have sharper p-values. So it's much easier if you look at like just doing it on the classically inbred strains by themselves. You get very broad and low peaks compared to very sharp peaks when you use on a Manhattan plot. You get very sharp and tight peaks when you use collaborative cross or HMDP. That's a very good reason for using that. Any others? Which mice are easier to care for? The classically inbred or the HMDP? Yeah. It is in fact the HMDP, because they're much better. They're much more robust, because they're not inbred. So there's a reason that there's people don't inbreed, because it causes bad things to happen. It makes you kind of less fertile, less active, lot of things. But generally the HMDP mice are easier to care for compared to just a classically inbred strain. Okay, any other benefits that people can come up with? Those are basically the two main ones. The collaborative cross HMDP mice, the ones that are more genetically diverse, are easier to handle. They also but in particular, you get a much sharper result. You get much sharper peaks compared to just using classically inbred strains. Okay, so that's oh yeah, question.
Sorry. So in the roundup is HMDP is always better because it's lower LD and then easier to care for.
So there are reasons why. So what are some reasons why a classically inbred strain might be better than an HMDP strain?
Easier to get.
Easier to get, right? It's you can just order these mice through the mail. Yeah.
Have been used more traditionally, so you can compare them back.
You can compare to previous studies. Yep, that's another benefit. These mice have been around for a long time, so there's a lot of literature on them. People know their behaviors. They know whether this mouse is smart or dumb, or whether it's nocturnal or diurnal. So there's a lot of literature out there for those mice, and it's easy to order, whereas it's work to do one of these studies, to do an HMDP study. Like when the Hurricane Sandy happened and a bunch of mouse labs got flooded at NYU, it took years to build up those mouse, lineages. So that's why it was very particularly tragic. Okay, any other questions on this mouse stuff? Great. So again, just some tips, like review the papers, the Flint paper, the Caviar paper. And for grads, there will be stuff on mathematical foundations. So you'll be asked to look at the matrices that represent genetic information and be asked to say things about them. But I'm not gonna put any proofs on there. I'm not gonna make you guys try and demonstrate why linear mixed models can be solved efficiently or anything like that. It'll be more of the mathematical foundations, but it'll be about as large in scope in terms of your grade as the question was on the midterm. So there'll be 25 points that are grad only. That'll be grads only. Yeah.
What do you mean describe the matrix? What should we kind of review?
So for example, look at the look at the math around mixed models. You're not gonna be asked to rederive it, but you should. So you're not gonna be asked to rederive anything, but you should understand it.
That's so the stuff that came shortly after the midterm, all the matrix math.
That's right. Yeah.
So the same physically in undergrad did a testable based on all those conceptual questions.
Yeah. So the test is going to be much more towards conceptual and like thinking about the stuff that you read and whatever. And this like anything that was on the slides, anything that was in the papers is testable. But as you guys know, a lot of what you just saw will actually end up on the exam. Yeah.
What will the paper questions be like? Because
I mean, they were like the ones that you saw. Like this is the questions here on like if you read the Jonathan Flint paper very carefully, like the answers to those are in here, or like from our discussion in class on mosh genetics, it's there as well. Yeah. Okay, great. That is that's it. This is going to go up on YouTube.